We are now on the last set of PowerPoints. We have Immunology Proper 2, wherein we will deal here antigens, antibody, the antigen-antibody reaction, and hypersensitivity reactions. So we will take up first what are antigens, what are antibodies, and the antigen-antibody reaction. Antigen is a substance that causes the immune system to produce antibodies against it. So it is considered to be any foreign substance from the environment, such as chemicals, bacteria, viruses, or pollens, and may also be formed inside the body as with bacterial toxins or tissue cells such as cancer cell formation, which are not recognized by the body. According to Adam, an antigen is a substance that induces the formation of antibodies because it is recognized by the immune system as a threat. So we have here an illustration of an antigen and antibody. So we have here the red blood cell. There are three characteristics of an antigen. We have immunogenicity, we have specificity, and memory. For immunogenicity, three characteristics are required. Must be foreign to the body, must possess a high molecular weight of 8,000 to 10,000 Daltons, must have a structural stability and complexity. There are certain substances in the body that cannot be considered as antigen because they do not possess high molecular weight. So we have the haptin, and for them to become an immunogenic cell, they require a carrier protein to reach up to a high molecular weight. For structural stability and complexity, the antigen must be made up of proteins and at least 30% immunogenic amino acids such as lysine, arginine, glutamine, and aspartame. The chemical components of an antigen could vary. They could be made up of polysaccharide. We have the glycoproteins and glycolipids. They could be lipids in nature. They could be nucleic acid in nature. And some of them are made up of carbohydrates and lipids, which are considered to be inferior antigens due to their simple and unstable structure, and nucleic acids are poor antigens due to rapid degradation. While antibody is a glycoprotein produced by the body's immune system when it detects harmful substances called antigens. The antibody is a Y-shaped protein on the surface of B cells or B lymphocytes that is secreted into the blood or lymph in response to an antigenic stimulus to neutralize the antigen by specifically binding to it. The antibody consists of two pairs of polypeptide chains, the heavy chains blue in color and the light chains pink in color. And they are arranged in a letter Y formation. The tips of the Y or the ends of the arms are the regions that binds to antigen and deactivate them. The Y formation of the antibody, we have the antibody consists of two pairs of polypeptide chains, the heavy chains blue in color and the light chains pink in color. And they are arranged in a letter Y formation. The tips of the Y or the ends of the arms are the regions that binds to antigen and deactivate them. The Y formation of the antibody, we have the arms and the stem. The end portion of the arm is the antigen binding site, and the stem end is the complement binding site or region. The arms and the stem are linked together by disulfide linkages, while the enzyme papain can cleave the antibody at the hinge region into two Fab fragments in the arm and one Fc fragment in the stem. So we also have the colors, the blue and the pink colors. The blue stands for the heavy chain and the pink stands for the lighter chain of amino acids. Then we have letter C for constant domain and V for variable domain. We mentioned about the variable region and the constant region. Between the two, the variable region is the antigen binding site. So the heavy chain is the constant region and the pink colored is the variable region or containing the light chain of amino acids. The light chain of the antibody has a molecular The light chain of the antibody has a molecular weight of 23,000 Daltons and contains 200 amino acids, while the heavy chain's molecular weight is around 50 to 70,000 Daltons with 400 amino acids and serve as bases for classification into immunoglobulin, or we have the gamete, also referred to as immunoglobulins. Gamete stands for immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E, and immunoglobulin D. So we have the different types of immunoglobulin. So we have the IgG, IgE, IgD, IgA, IgM. And we can see the complexity of the structure of the immunoglobulin or the antibodies. The Y formation of the antibodies could be monomer, dimer. We have the three chains and we have the pentamer formation. So we have, for the monomer, we have IgG, IgA, IgD, and IgE. And for the membrane-bound monomer, we have the IgM. The IgA is an immunoglobulin having two Y formation. We have the dimer, and IgA can also be referred to as secretory dimer. And we have the IgM, which is a pentamer. Immunoglobulin G is the gamma immunoglobulin. It has a molecular weight of 150,000 Daltons. It is involved in the body's main defense against bacteria and makes up 70 to 75% of total human Igs. And it is the most abundant form in the globulin fraction of blood plasma. And the IgG is the only class that crosses the placenta and considered to be the most versatile, very effective in establishing humoral immunity. So take note, the only immunoglobulin that crosses the placenta. So based on the illustration, it is a monomer. So in continuation, IgG is found in cord blood and CSF. It activates complement and important in opsonization. The phagocytes or the phagocytic cells have receptors for Fc fragment of the IgG. The baby's serum is mostly maternal immunoglobulin at birth or after birth. And this IgG will increase in concentration between 3 to 6 weeks after birth. And there are four subclasses or units. We have the IG1, IG2, IG3, and IG4. So the IgA is the alpha immunoglobulin, the second most type of immunoglobulin in the body, around 15 to 20% of immunoglobulin. They are found in the membranes of the upper respiratory tract and in the digestive tract. They are also found in tears, in saliva, in the mucus, and colostrum. We have two subclasses of IgA. We have IgA1 and IgA2. So based on the illustration, IgA is a dimer. SIG-A stands for secretory immunoglobulin A. So this type of immunoglobulin protects body surfaces from invading pathogens by interfering their adherence and colonization. IgM is the mu IgM is the mu immunoglobulin having a molecular weight of 900,000 Daltons, and we have 10% of immunoglobulin pool in the body. And they are responsible for blood infections and the first antibody produced by the immune response. So take note of this. First antibody produced by the immune response and help trigger additional production of IgG, and they are also present on lymphocytes, and they clump microorganisms, specifically gram-negative bacteria. So based on the illustration, IgM is a pentamer. There are 5 Y formation of the antibodies. Immunoglobulin M is the first immunoglobulin expressed in the embryo, present together with immunoglobulin D on the surface of resting lymphocytes in postnatal life. It serves as receptor for antigen and transduces the signal for proliferation and secretion of antibodies early in an immune response, but it is soon replaced by immunoglobulin G. IgM is efficient in fixing complement, lytic reactions, and enhances phagocytosis. And it consists of 5 subunits referred to as pentameric. So IgM is a pentameric immunoglobulin. IgM is also involved in complement fixation reaction. A complement is a group of blood proteins which, when activated, cause a cascade of enzymatic reaction that result in chemotaxis, phagocytosis, and lysis of foreign cells and bacteria. IgE or immunoglobulin E is epsilon immunoglobulin. It has a molecular weight of 72,000 Daltons, and they are found in mucous membranes and skin. Least antibody found in blood serum, it is only around 0.004%. The IgE is heat labile and triggers some hypersensitivity and anaphylactic reactions involved in parasitic infection, specifically the helminthic parasitic infection. The Fc region of IgE binds strongly to mast cells and basophils. Together with antigen, the IgE mediates the release of histamine and heparin. IgD is the delta immunoglobulin. So based on the structure, it is monomeric. It has a molecular weight of about 180,000 Dalton, and they are present in serum in tiny amounts or in tiny concentration, about less than 1%. And they are found on surface of B cells in association with IgM or immunoglobulin M. And they are susceptible to proteolysis. IgD is used as receptor for antigens, and they also help to anchor to cell membranes with its abundance of amino acids. Not completely determined why IgD is found in serum, and this is the least understood antibody. We have here we have here an illustration of antibody-antigen binding. We have here an illustration of antibody-antigen binding. So the Y formation is the antibody, and the light brown structure is the antigen. The surface of the antigen contains the epitope or the receptor site, and which will react to the antibody at the tip of the arm. So, for the antigen antibody determinant sites or sites of reaction, we have for the antigen, we have the so for the antigen antibody determinant sites or sites of reaction, we have for the antigen, we have the epitope, And for the antibody, we have the paratope. So take note, epitope for the antigen, paratope for the antibody. We have here an illustration of the different shapes of the paratope of the antibody and different shapes of the epitope of the antigen. So the yellow colored structure are the antigens and the blue structure are the antibodies. There are antigens that are described to be multivalent antigens. So these are antigens that contains different epitopes. So they have more than one epitope and will react to an antibody depending upon the epitope of the antigens. Usually, these are microorganisms like the bacteria that are very pathogenic due to the different types of epitope it possesses. We have here an illustration to differentiate. We have here an illustration to differentiate a multivalent antigen with different epitopes and multivalent antigen with repeated epitope. So in a repeated epitope, the same shape of epitope is present in one antigen or antigenic cell. While on the other side, we have an antigen or antigenic cell having several shapes of epitope. So it can react to different types of antibodies. How do we know that there is antigen-antibody reactions? The reactions are manifested in the laboratory analysis of the antigen-antibody reaction in the form of agglutination, precipitation, flocculation, and complement fixation, and some specific immunoassays. There are instances wherein an abnormal type of response is formed. We call this as hypersensitivity reaction, and this is an abnormal immune response of the body, or it is described to be an immune response gone awry, and further described to be an exaggerated and harmful immune response due to interactions between antibodies and allergens, so commonly referred to as allergic reactions. These require a pre-sensitized immune state of the host. We have here a table for the different types of hypersensitivity reactions. So we have mnemonic here, acid. On the first column are the types of hypersensitivity reaction, and second column is the description or what causes the hypersensitivity reaction. So we have type 1. This is immunoglobulin E mediated, quick onset after exposure, and commonly referred to as allergic hypersensitivity reaction. And this is due to bee stings, may be due to latex, or may be due to certain medication like antibiotic and other drugs. Then type 2 hypersensitivity reaction is cytotoxic antibody-mediated hypersensitivity reaction, or simply referred to as cytotoxic hypersensitivity reaction. And this is due to hemolytic reactions, good pasture syndrome, and hyperacute graft rejection. Type 3 is the immune complex or IgM mediated hypersensitivity reaction, or simply referred to as the immune complex deposition, due to hypersensitivity pneumonitis, systemic lupus erythematosus, due to polyarteritis nodosa, and serum sickness. Then type 4 is the delayed or cell-mediated hypersensitivity reaction, or simply referred to as delayed hypersensitivity reaction. So due to chronic graft rejections, PPD test, latex, nickel, and poison ivy. So if we will go through the different types of hypersensitivity reaction, we have acid. We have mnemonic acid. A for allergy, C for cytotoxic, I for immune complex deposition, and D for delayed hypersensitivity reactions. For type 1 hypersensitivity reactions, we have several allergens in the environment causing this type of reactions. I will give you few seconds to view the different allergens for type 1 hypersensitivity reaction. And together with the allergens in the last slide, we also have spores coming from the fungal organism that causes type 1 hypersensitivity reactions. We have here steps involved in allergic reaction. So it is numbered 1, 2, 3, 4, and 5. So for number 1, we have initial allergen exposure, and number 2, we have the involvement of the immunoglobulin E, so antibody IgE production. Then number 3, we have histamine producing mast cell, which are also involved in this type of allergic reaction. So from step 3, we have the immunoglobulin E found on the mast cell. So the mast cells are sensitized with the IgE. Then followed by exposure to the allergen, which causes the release of the granules in mast cells, causing allergic reactions. We have here an illustration of the interrelationship of IgE with the granules of mast cells causing type 1 hypersensitivity reactions. The most common type of signs and symptoms of type 1 hypersensitivity reaction is sneezing, just like here in the illustration. Other signs and symptoms of hypersensitivity reaction involves the skin, the respiratory, the gastrointestinal tract, the cardiovascular system, the neurological system of the body. So under each, we have the different types of signs and symptoms. You just read through the different signs and symptoms under each category. So aside from sneezing, the most common type of sign and symptoms is the presence of swelling on the surface of the skin we call hives. I think this is the last slide. So this end up immunology, but I would like to recommend further readings about immunology. Thank you for your patience.